morning students. So today we are going to discuss about RNA. So RNA, all you know that it consists of ribose nucleic acid and the alternate phosphate molecules, and there are four bases are there: adenine, guanine, cytosine and uracil. The thymine base which is there in DNA, so that is not present in RNA. In place of the thymine, the base is uracil. Okay. So there are three different types of RNA: ribosomal RNA, messenger RNA and transfer RNA. So the RNA that is a single strand molecule, as the DNA that is a double strand molecule, the RNA that is single strand, and if the RNA is becoming double strand, then it can lose its biological property. Ribosomal RNA, first we will discuss about ribosomal RNA. So what is the function of the ribosomal RNA? The ribosomal RNA that is found in ribosome, and it helps in the formation of ribosome. Then the messenger RNA, so that the function of the messenger RNA is to transfer the message or the information from DNA to the site of protein synthesis, that is ribosome. So it is getting the information from the DNA that what kind of protein will be synthesized. So as it is carrying message, the name is given messenger RNA. Then the transfer RNA molecules, they can transfer the amino acid from the cytoplasm into the site of protein synthesis. So that is the messenger RNA and the ribosome. Okay. So these are the primary function of ribosomal RNA, messenger RNA and transfer RNA. Now the first RNA, which is the ribosomal RNA, as I told you, so that is found in ribosome, and it comprises 80% of total RNA. There are three types of RNA. So out of these three types of the RNA, it is 100%, then the 80% consists of ribosomal RNA. And in prokaryotes, it is formed on a part of DNA called ribosomal DNA. As you know that in the process of transcription, there is formation of the new RNA molecule on DNA strands. So the part of the DNA which is making the ribosomal RNA, so that part of the DNA is called ribosomal DNA in prokaryotes. Whereas in the eukaryotes, it is formed on the DNA which is present in nucleolus. There is difference between nucleus and the nucleolus. Nucleolus, so that is located inside the nucleus, and inside the nucleolus also the DNA is also present. So in case of the eukaryote, it is formed on the DNA which is present in the nucleolus. Okay. So either it is present as in the form of the straight rod, or you can see the coiling, so that is present, or in the form of the extended form. The second type of RNA, that is messenger RNA, and the name messenger RNA, so that was proposed by Jacob and Monod. It is always single stranded and each gene transcribes its own messenger RNA. तो messenger RNA जो है ये नेम इन्होंने प्रपोज किया था, जैकोब एंड मोनोड, और messenger RNA हमेशा सिंगल स्ट्रैंड RNA है, और इसका काम क्या है, ये DNA से इंफॉर्मेशन ले रहा है और ये इंफॉर्मेशन दे रहा है कि किस तरह का प्रोटीन का फॉर्मेशन होना है. ओके. Now these are all the different parts of messenger RNA. So at the 5' end of messenger RNA, because the RNA, so that is also made up of the ribose sugar, and the ribose, so that is a 5-carbon compound. So here the 5th carbon, so that is exposed, so the end is called 5' end. So at the 5' end, there is one cap is present, and then what is there in the cap? In the cap you can find out methylated guanosine. Guanosine, which is attached to a methyl group, so that is present in the starting position of the messenger RNA, and that area is called cap. Then after cap, you can find out there is another small area, and that area is called non-coding area. So non-coding means it is not giving any kind of information. So the name is given non-coding. Then the initiation codon, the first initiation codon, so that is A the initiation codon, so that is constant and it is AUG. Then a large area, so that is called structural messenger RNA. So it is the coding area, it is giving all the information what kind of a protein is to be synthesized or produced. Then the next, there is termination codon, and the termination codon is also known as the stop codon. Process will stop. When the codon will reach, when the ribosome will reach to the codon, then the protein formation process will stop. And there are three termination codon: UAA, UAG, and UGA. Then after that, there is a small area which is called again the non-coding area. It is not giving any kind of the information. And at the 3' end, there is a long poly A sequence, just like A like this. So at the 3' end, you can see there is a long sequence of only the adenine base. Okay, so this is about the structure of messenger RNA. The next type of RNA, so that is transfer RNA. Transfer RNA is also known as the adapter molecule, or it is also known as the soluble molecule because it is too small to be precipitated. So the name is given soluble RNA. And the primary structure of transfer RNA, so that was worked out by Holly et al. In 1965. And the model which was proposed. So this is a very well-known model which is explaining the structure of transfer RNA molecule. And the name of the model is clover leaf model. Clove मतलब लवंग. तो लवंग जैसा इसका आर्म दिख रहा है. One stalk is there and one spherical structure is there. So the name is given the clover leaf model. Okay. So as per this model, there are 5 arms of transfer RNA molecule. देखिए हर RNA molecule जो है ये single strand है. या तो ये सीधा है जिस मैसेंजर RNA में है, या फिर ये मुड़ा मुड़ा coiling form में भी आपको मिल सकता है. ठीक है. तो यहाँ पर 5 आर्म है. The first arm, so that is called acceptor arm. This is the 5' end of ribosomal RNA. This is the 3' end. And the 3' end at the starting position there is CCA. So that is present. And 3' in third carbon of the ribose, which is having the OH functional group, so that is exposed. Okay. So this acceptor arm, so that is the arm which is actually combining with the amino acid. क्योंकि ट्रांसफर RNA मॉलिक्यूल जो है उसका फंक्शन क्या है? ये जो फ्री अमीनो एसिड जो है साइटोप्लाज्म में, ये उसके साथ कंबाइन करके उसको पिकअप कर रहा है और कहाँ ला करके छोड़ रहा है मैसेंजर RNA के पास, जहाँ पर प्रोटीन का फॉर्मेशन हो रहा है. तो अमीनो एसिड कौन सा आर्म से जुड़ेगा ट्रांसफर RNA का? सो दैट इज द 3' end. और पहला यहाँ पर ग्रुप जो है ये हमेशा कांस्टेंट है CCA. तो ये कार्बन, तीसरा कार्बन का जो OH फंक्शनल ग्रुप है उसके साथ अमीनो एसिड जुड़ रहा है, इसलिए हम उसको बोल रहे हैं एक्सेप्टर आर्म. और ये 5' end में या तो G रहेगा या फिर C रहेगा स्टार्टिंग पोजीशन में. ठीक है. नाउ द सेकंड आर्म, सो दैट इज कॉल्ड द D आर्म. इसका काम क्या है? यह अमीनो एसिड को रिकॉग्नाइज करता है, क्योंकि आपका साइटोप्लाज्म में बहुत सारे बायोमोलिक्यूल्स रहेंगे. अमीनो एसिड रहेंगे, कार्बोहाइड्रेट रहेंगे, प्रोटीन रहेंगे और दूसरे तरह के बायोमोलिक्यूल्स रहेंगे. तो इसका फंक्शन है कि ये अमीनो एसिड को पहचान रहा है, रिकॉग्नाइज कर रहा है. ओके. Then this is called the anticodon arm. Anticodon arm, so that is having 3 nitrogenous base. And 3 nitrogenous base, as I told you, so that is making 1 codon. For example, the initiation codon AUG. ठीक है. तो ये एंटीकोडन आर्म जो है ये मैसेंजर आरएनए के जो कोडन है उसको रिकॉग्नाइज करेगा, क्योंकि आपको मालूम है कि आरएनए जो है उसका बेस जो है ये डीएनए का कॉम्प्लीमेंट्री बेस है. ठीक है. लाइक फॉर एग्जांपल, सपोज द बेस ऑन द मैसेंजर आरएनए, सो दैट इज AUG. तो एंटीकोडन क्या होगा? A की जगह में U, थायमिन नहीं है, U की जगह में A और G की जगह में C. So this is anticodon and this is codon. तो कोडन एंटीकोडन जो है एक दूसरे को रिकॉग्नाइज करते हैं, उनका बेस भी कॉम्प्लीमेंट्री है. उसके बाद आपको एक छोटे से आर्म मिलेगा जिसका नाम है वेरिएबल आर्म. इसका कोई पर्टिकुलर फंक्शन नहीं है. ठीक है. इसमें नंबर ऑफ बेस जो है ये वेरिएबल है. उसके बाद T साइज C आर्म जो है ये राइबोसोम रिकॉग्निशन साइट, ये राइबोसोम को रिकॉग्नाइज करता है, क्योंकि जब प्रोटीन फॉर्मेशन हो रहा है तो राइबोसोम जो है मैसेंजर आरएनए के साथ बाइंड कर रहा है, तो ये आर क्या कर रहा है, रिकॉग्नाइज कर रहा है राइबोसोम को और वहां लेकर के अमीनो एसिड जो है उसको छोड़ रहा है. So this is about the structure of transfer RNA molecule. Now the question is, what is transcription? Transcription is the process of copying genetic information from one strand of DNA so that one new RNA molecule can be synthesized or produced on DNA. So the DNA which is responsible for transcription, so that is called, that is the non-coding strand. So that is called non-coding strand, not the coding strand. Okay? Or transcription unit. The DNA which is taking part in the process of transcription, it may be called as transcription unit. So as in the replication process, the whole DNA is replicated. In transcription, only a part of DNA is taking part. Either one gene is transcribing or a small part of the DNA, so that is taking part in the transcription. So that is the difference between the transcription and the replication. In replication, on DNA, new DNA molecule is produced. In transcription, on DNA, a new RNA molecule is produced. In replication, both the strands of DNA they are participating, whereas in transcription, only one strand of the DNA, so that is participating. Okay? So here also you can see the base, they are complementary. For example, in board you get this kind of a question. For example, this is the DNA, and on DNA, the transcription process is going on. Okay? For example, suppose the base are given. So this is the base which is present on DNA. Now the question will be given, you write down the corresponding bases in the new RNA molecules. Okay? So thymine, so that will pair with the adenine. Again the thymine, so that is paired with adenine. Adenine will pair with uracil because there is no thymine base. So here will be U. Then cytosine will pair with the guanine. Guanine will pair with cytosine. Then again cytosine. Cytosine is pairing with guanine. Adenine is pairing with uracil. Thymine is pairing with adenine. So this is DNA. And this is the corresponding base in RNA. So like this you will get the question. Okay? Now the question is, as I told you, in transcription process, out of two DNA strands, only one is participating. Why? So this is a very important question. Okay? Now you see this diagram. This is a DNA molecule, and when it is participating in the transcription process, now it will open, the bond will break, it will open, and now this is a single strand of DNA, this is a single strand of DNA. Okay? Suppose both the strands are participating in transcription. Okay? So here I have written the base. The base on DNA is P, C, G, A, T. And this is the new RNA molecule which is produced. The corresponding base will be T will pair with A, C will pair with G, G will pair with C, A will pair with U, again the A with U, and T with A. Okay? So now here I have written all the base of RNA molecule A, G, C, U, U, A. I told you three base is called a codon. One codon can specify one amino acid. So A, G, C is one codon, U, U, A is one codon. Okay? Now if this strand is also participating in transcription, direction will be this. Here the direction is downward, here the direction is upward. And the basis of the new RNA molecule here I have written starting from this direction A, G, C, U. So here you see the same strand of DNA. Now that is making two different type of protein. Now this codon can specify one amino acid. This codon can specify another amino acid. Here you see the codon is different. Now it can code for another amino acid. It can code for another amino acid. So if the same DNA will prepare the two different type of the protein, then the process will be complicated. Okay, so that is the reason why out of two strands of the DNA, only one strand is participating in the process of transcription.